Okay, just welcome to everyone. As you know, Ben and his partner Gemma actually came to us from the UK a while back, in much better times, I might say. And keeping it in the family, both of them have been interested in coral reef pinnacles and sea mounts. I think because they really are like the biodiversity hotspots of coral reefs and just incredibly interesting places. Ben's thesis, when he's finally allowed to actually reconnect with his study site, which doesn't look like it's happening anytime soon, but it will be on connectivity. So he's really interested in how fishes on these sea mounts and pinnacles sustain their populations and how they interact with one another and so on. But I'll leave that to Ben to explain to you. So I'll turn it over to Ben now. Perhaps I could just remind everybody, probably at this stage, to turn off your sound, your microphones and your cameras, just so you can focus on Ben's presentation, and then we'll just open it up for questions at the end. So I will tune out, and it's over to you, Ben.
All right, thanks, Jeff, and thanks everyone for zooming in. It seems like a pretty appropriate time to be discussing things like connectivity, given the existing situation that's going on around us and how connected we are all, unfortunately, at the moment. But hopefully this will be a bit of light relief for everybody. And so, subject of my thesis is sea-state connectivity in fishes on coral reef pinnacles. So, just going to kick off with the idea of what is ecological connectivity. And essentially, this is an incredibly broad concept with applications all across ecology. And for that reason, there are a whole heap of definitions that surround the idea of ecological connectivity. But most of them are to do with movement and linkages of things like organisms, energy, nutrients, and other chemicals across different patches of habitat. And there's one definition that encompasses that. Again, things like physical events and materials, organisms moving around in space and time, connecting habitats and populations. For my purposes, I'm going a bit broader than that with my definition, and I'm just going to consider it as the strength of the biological, chemical, and physical linkages within a natural system. While there is limited consensus about the definition of connectivity in ecology, it's pretty typical to find an organism-centric approach, and specifically looking at how organisms link ecosystems. And there are a whole heap of examples around this. And we've got things like dispersal of larvae and seeds and the like, foraging activity of animals and how that connects systems, ideas around ontogenetic movement and life history changes and how these connect habitats and populations, migrations of adult individuals, things like trophic cascades, trophic relays that link food webs, and then there are also a whole gamut of things around nutrient transfer and hydrology that we do need to bear in mind. Why is it important? Well, it's important because it drives ecological paradigms. And specifically, spatial linkages are the things that really determine the distribution and movement of organisms and the underlying ecology. And connectivity gives us a means to quantify the dispersal of things like propagules and materials and nutrients through the systems in which they're found. Very importantly, the strengths of linkages may be really important in determining how systems and populations can recover from disturbance events, or it may have the opposite effect. Strong linkages may allow the distribution of disease and pollutants through systems and things like invasive species. Connectivity has implications for management, as a lot of you will be familiar. It can be used to inform effective spatial protected area design, so how we lay out reserves in nature can take into account connectivity. And it may help us understand how best to conserve particular species or subpopulations. And generally just maximize the efficiency of management effort and increase the payoff of our dollar investment into conservation. And just important to remember that connectivity occurs at every single level of ecological organization. Okay, so where does it come from? How did it evolve as a discipline? So really, if we could find the roots of modern connectivity in a whole bunch of preceding theories, most of which started in the terrestrial environment with things like island biogeography theory and the idea of metapopulations, which cropped up in the 60s. In the 80s, these terrestrial ecologists brought us ideas like habitat patch dynamics and connectivity using movement corridors through woodlands and the like. But the marine environment's had all of its analogous theories over the years. We've got concepts like blue corridors, which cropped up in the early 2000s. Things like trophic linkages, which have been heavily studied in the marine environment. Clearly, larval dispersal is a big one from our lab group and the like. And ontogenetic migrations, which are also being heavily studied. Okay, and this is reflected in systematic literature searches. We have things like, so systematic literature search. So looking at things like landscape and marine connectivity over the years, how these have really influenced things. So we've got this whole period of time sort of before the 1990s where we really didn't see a whole bunch of work, but there were things like habitat patch connectivity theory getting formed at this time, movement corridors. These theories were sort of coming online at about this time. And then building up to things like theories of larval connectivity on the Great Barrier Reef. Callum Roberts, 1997, modeling larval dispersal around the Caribbean, hydrologic connectivity theory in the early 2000s, and this really leading to like a massive explosion of work over the 2000s in connectivity. How that applies to in sort of the wider context, you can see really here that was the landscape and the marine connectivity figure that you just saw then. In the context of marine ecology, really there was only a little bit of work going on in that sort of area from the 60s right through to the late 80s, with about 30 papers per year on ecology. And then we see a really big boom in marine ecology theory taking off from about 1990 onwards. But connectivity is not far behind. And in the modern era, from the results of my literature research here, we've got about 14% of all marine ecology theory is comprised of connectivity research. So when we're looking at connectivity, we're really thinking about three overarching categories. And the first is structural, which is essentially the idea that you can infer connectivity metrics about just the physical and the geographic features of a system. Things like isolation and barriers within in a system would be the kind of thing we're talking about there. Secondly, it comes functional connectivity, which starts integrating biological information about a system. So life histories and the underlying ecology starts getting taken into account to build up an idea of how things are functionally connected. And then realized connectivity, which is really all about the empirical observations and measurements of linkages in nature. And for the second of those two, functional and realized connectivity, they're really the end goals of ecologists in this context. And within that context, fishes make really good model organisms. And using fishes, we can investigate a whole range of connectivity types using a wide variety of methods. And some of those include things like movement and migration of organisms, which gives you a measure of realized connectivity. And we can use things like biotelemetry, tagging and tracking to track organisms through the environment. Population and larval connectivity measured by dispersal, and it gives you an idea of realized connectivity, and the kinds of tools we can use for that are things like genetic parentage analysis. And then the idea of trophic or ontogenetic connectivity, which gives us insights into how habitats are functionally connected, and we can use techniques like stable isotope analysis to get some measures of these. Okay, application. How's all this work rolled out for use in the real world? Well, there's an interesting study that came out last year that specifically looked at the uptake of ecological connectivity research into protected area design. And what these guys found was that out of 186 studies that made recommendations for 746 marine protected areas, there was nothing before the year 2000, so reinforces that idea of connectivity being a recent discipline. A big increase since 2010. And they wanted to know really whether this was reflected in protected area design. And what they found was that only 11% of MPAs in their study actually utilized the recommendations from the studies themselves. And within these, this 11%, 71% of them were found in California or Australia. So the upshot of that is that there's bad and uneven uptake of connectivity research globally. So that's a bit about the application. What about knowledge gaps? So that brings me on to my study system and study species, which is fishes and pinnacle reefs. So when it comes to pinnacle reefs, for those of you that are not familiar with these are relatively small, isolated, submerged habitat patches, usually very isolated and remote from the rest of the system, and they can contain large aggregations of things like mobile predators and planktivores, and also home to a number of charismatic species. And really there's probably a whole heap of unique paradigms that occur on these systems that we don't really know about just yet, and so that's kind of why they're of interest to me. So moving that into a schematic of a generalized pinnacle. As I said, we're talking about these isolated submerged structures, and they rise from the ocean floor, typically hundreds of meters deep, and rise up into the euphotic zone, so within, say, around 10 meters or so of the sea surface. Okay, they may be small, but they're ubiquitous. There's no consensus about how many pinnacles and seamounts there are in the world. And this is a bathymetric map from the Tasman Sea, and you can see just off the east coast of Tasmania here, there's just a whole heap of these structures present. Early research from our lab group is showing that they contain unique assemblages of fishes, at least, and these two figures here, this multi-dimensional scaling is showing some interesting differences in community structure with pinnacles as compared to offshore emergent habitats nearby, and also nearshore reefs. And for those same three groupings, pinnacles also have a unique and distinct group of fish species themselves, 75 species there. Okay, and what do we know about these small systems? Are they well studied or are they ignored? And a recent review by Gemma in 2018 basically found that when you look at coral reef systems as a whole, including pinnacles and coastal systems, nearly 27,000 of the publications that were returned by that search were on coastal systems in comparison to only a few hundred on seamounts and pinnacles and other remote systems. So there's a really small amount of work conducted on these pinnacle systems. But then how does that translate to work on connectivity? Well, unfortunately, that's kind of pretty much the same case. So a review conducted in 2017 found that nearly 600 papers on connectivity in the context of marine systems were on reefs and coasts, nearly 100 on the pelagic systems, and then only 20 on seamounts. So yet again, we're seeing that seamounts and pinnacles are largely excluded from a lot of these connectivity studies. The other thing that this review found was that there's a very limited amount of investigations across ecosystem connectivity, or cross habitat connectivity, which is things like the land-sea interface and things like that. Okay, so how important are these pinnacles in connecting with the wider environment? Well, there are a few ways in which we think that these habitats might be linked. So here's our generalized pinnacle in a wider seascape. And clearly, one of the ways that they can be connected is through the dispersal of larvae, inferring genetic connectivity. And obviously, we can use genetic parentage analysis to delve into some of that. again, movements and movement connectivity, inferring some realized connectivity into the system, and we can use biotelemetry and tagging and tracking to measure some of that. And then trophic and ontogenetic linkages also may be going on, and tools available for that are stable isotope analysis and geochemical approaches. These are generally well-researched areas, so most of you will be familiar with a lot of these techniques and a lot of studies in these areas, but not on pinnacles. These are really very understudied systems in the context of these types of connectivity. And also these multiple method approaches are not really used on one, they haven't really been used on one study system so far. So that's part of my aim in this present study, is to do that. Okay, getting on to the study site then. So some of you will be familiar with this location. It's Kimby Bay in New Britain in Papua New Guinea, and it's located just up here off the main island. And Kimby Bay is located on the north coast. It's an embayment of about 25 to 30 kilometers across, and it's basically surrounded by this volcanic ring. And if you look on this map, this bathymetric map here, now we're looking towards the southwest. And green is terrestrial environment, so those are sort of the mountains surrounding the bay. And then that topography is carried on into the underwater area in blue. And you can sort of pretty clearly see some of the features of this caldera that sweep around the bay like so. And this is where we find a lot of our sites. So Kimby Bay has been used in the past a lot for studies on nearshore sites, and so it gives us a good opportunity to make a comparative study here with the pinnacles. So here's the locations of some of the nearshore sites that have been well studied by our lab group, at least for a couple of decades. We also have a whole bunch of offshore emergent sites. So these are offshore sites that come up to within a few meters of the surface. And these are also well studied by our lab group. And then we have the pinnacles located remotely 10 to 20 kilometers offshore. And these are really the focus of what I'm trying to get at. My project approach is I'm basically using a few approaches to investigate a number of aspects of connectivity. So the first is trying to get at some larval dispersal information to investigate realized connectivity. As you know, these genetic methods are well utilized for larval dispersal work, and again by our lab group, even in Kimby Bay. Lots of studies, but nothing yet on pinnacles. And so we don't really know anything about whether organisms on these habitats, whether they self-recruit, whether they are sinks from elsewhere or sources of larvae for other locations. And we hope to be able to conduct a temporal component on this and see whether dispersal and self-recruitment changes over time. Second approach is to investigate predator movements and whether this infers any realized connectivity. Using some biotelemetry methods, which really is kind of the gold standard for measuring realized connectivity, and come up with some ideas about how these pinnacles are connected from these movements. Predators we know are abundant on pinnacles, and this is some initial data that we've got so far from, we've teased out from data we collected in 2018 and 2019, which shows that on the pinnacles, predators are much more abundant, and so they're a good candidate for this kind of work. And then looking at stable isotopes to investigate some functional connectivity as a result of life history migrations. This figure here is an example from the GBR of a snapper species, and how through its life history migrations, it sort of forms this blue highway that connects nearshore habitats with the offshore reefs. So that's the kind of thing that I really want to do with these pinnacles, and see whether there's any kind of connectivity like that with the nearshore environment. And so I can use this stable isotope approach to track nutrients through food webs over time and space. Okay, research aims, the overarching aim is to pull all this together into one multidisciplinary cross-habitat study, and hopefully develop our understanding of the ecological role of these pinnacles across the seascape of Kimby. Doing that, firstly, I'm going to build on the existing connectivity work through parentage analysis, using a similar approach to investigating larval dispersal on the pinnacles. Secondly, I'm using movements of key mobile predators to see how these might confer realized connectivity with the wider seascape. And then looking at some of the trophic and ontogenetic linkages that connect the pinnacles with other habitat types. And as a result of all this, I really do hope that we can generate some useful information to help the local and regional government plan and incorporate these pinnacles into their management plans. Okay, so the general outline of my thesis. After the general introduction, Chapter 1 and Chapter 2 are together going to deal with the parentage analysis, with Chapter 1 being the microsat marker development, and Chapter 2 being the connectivity work itself. Chapter 3 is going to be a telemetry study to try and ascertain some information about key predator movements and whether these provide realized connectivity with the rest of the bay. And then Chapter 4 is getting at some of these ontogenetic migrations of similar mobile predator species, and whether those actually infer any functional connectivity to the rest of the bay. And then finishing up with some general conclusions. So just a bit of an overview of Chapters 1 and 2 together, because they are so connected and linked together. So this is going to be the genetic parentage analysis together to assess larval dispersal in a model reef fish on the pinnacles. And as I've mentioned before, this approach has not yet been used on pinnacle reefs, but these systems are really particularly well suited for this kind of approach, because they're small, remote, isolated habitats. They're just like a needle in the middle of the ocean, and so even though they are in that sort of isolated type of a scenario, they are able to sustain really highly diverse and high biomass communities. So why is this? What can what paradigms are at play here to allow this to happen? And finally, we were able to, on these small sites, sample the whole population. Because of their small size, we can really get around, for certain species, we can get around and sample the entire population of that species and really get an idea of what parent-offspring relationships are going on. There's a whole heap of modern genetic methods available, and we're going to be going with microsatellites due to we've got a lot of experience working on these in our lab group, and they're pretty easy to use, but there are other options available. We're just going with microsats. And the other sort of thing to note is that the development of the microsatellite markers is a prerequisite for the actual parentage analysis itself. So that brings me on to Chapter 1 in specifics, and this is going to be the development of the marker panel. So the research aim is to develop 20 suitable loci for use in subsequent parentage analysis. And what we're looking for here is high degree of polymorphism in these loci, low error rates, and conformity to Hardy-Weinberg equilibrium. The study species, as before, Amphiprion peridoreum, the pink skunk anemone fish. And really the reason we've chosen this is that it has a pretty suitable life history. It forms groups that are resident in anemones, which we can tag and locate, and we can go back to them over and over and over again. It's straightforward, therefore, to sample the whole populations of these things, and they also find counterparts around the bay, so conspecific in the rest of the some nearshore habitats. Okay, data collection then. So it's pretty straightforward field work for this. Basically go out on a short trip and collect 30 individuals, starting at the pinnacles themselves, which we'll fin clip. Those fin clip samples are stored in ethanol and returned to JCU for DNA extraction. Once we've got the DNA extracted, then we're going to move the whole operation to a partner lab in King Abdullah University of Science and Technology in Saudi Arabia. And there we're going to develop the markers as per existing methods. There's a few guidelines that we'll be following. And obviously the anticipated results that we're after is a panel of markers that we can use in Chapter 2, which brings me on to Chapter 2 nicely. And that's the larval dispersal study itself. So research questions here, there's a few of them. The first and foremost is, to what degree do these organisms self-recruit on the pinnacle reef? So do they spawn and their larvae just all head out into the blue somewhere, or do they self-recruit back onto the reefs? Second question is whether there's any connectivity between these pinnacles. And then the third one is expanding the idea out to the wider seascape connectivity and seeing whether the pinnacles are connected through larval dispersal with some of the nearshore and offshore habitats. Just a reminder of the study species, and then data collection and the sampling design. So we've got these 4 pinnacles that we're going to be working on that are laid out in this map here on the right. And on these, we're going to conduct exhaustive sampling of all groups of Amphiprium peridoreum. And we'll fin clip the adults and remove the recruits for DNA extraction. And then the idea is that we'll repeat this over time, as I mentioned earlier, really getting an idea of whether this self-recruitment or lack thereof is consistent over a time period. And then once we've done that, we'll be expanding out to the surrounding reefs to really drive at that question 3, whether there's any connectivity with the wider seascape. Analysis, much the same as for chapter 1. Samples extracted and brought back, samples stored and brought back to JCU for DNA extraction. The fragment analysis also conducted at KAUST, and then microsatellite scoring using the marker loci that we developed in chapter 1. Okay, scoring is going to happen. We're going to use a program called GeneMapper, and there's also a package in R called MSAT allele, which gives us those sorts of outputs. And then once we've got that, we'll be identifying parent and offspring pairs using a maximum likelihood approach in this particular piece of software called FAMOS, and that will enable us to assign potential parents to offspring. Okay, and then the end goal is obviously to identify dispersal patterns and measure degree of self-recruitment and whether there is any realized connectivity with the wider environment. Okay, that's chapter 2. Moving on to chapter 3. So away from peridorean and on to the predators, the mobile predators, and whether they infer realized connectivity on these pinnacle reefs. So just an overview of what we're going to be doing here. So this component is all about tracking and tagging and tracking pinnacle predators and making empirical measurements of their connectivity as they move around the seascape. And as they move around the seascape, they may infer realized connectivity for the pinnacles with the rest of the habitats in the bay. Research questions. Again, there's a few. Firstly, what I really want to get at is whether, for select mobile predator fishes, whether if on a pinnacle, whether they actually have a higher site fidelity than on an extended reef, with the rationale being that when you've got more territory available, do these species actually spread out and start using these extended nearshore reefs compared to the pinnacles, where I think they're probably going to be sticking around in one spot. For species that we tend to find almost exclusively on the pinnacles, which I'm calling pinnacle specialists, I really want to know about whether there's any inter-pinnacle migrational movement, or even if they move away from the pinnacles at regular intervals. So, for example, if they move away at night and go foraging for something else somewhere else, this gives us a bit of a window into things like energy subsidies that might be going on to these pinnacles if they're feeding elsewhere and bringing things back. And then for the species that we tend to find everywhere on pinnacles and on nearshore environments, I really want to know whether these generalists display more site fidelity on the pinnacles in comparison to the extended nearshore reefs, and whether they have smaller home ranges. Species selection. So I've got 2 mobile predator fishes that I'm interested in here. The first, like I said, I'm calling a pinnacle specialist because we really only tend to find these on the pinnacle reefs in large schools like the one you see here, and that's the chevron barracuda or Spirina caney. And then what I'm calling the habitat generalist, because we find them on the nearshore and the offshore and the pinnacle reefs, is the bluefin trevally or Carex melonpygus, and these tend to hang out either individually or in small groups of 2 to 4 individuals. Okay, back to the map and the study design. For this project component, I've gone with 3 separate receiver arrays, and they're as follows. So the first is the pinnacles themselves, and really each pinnacle can only accommodate 1 receiver. So there's 3 pinnacles that I'm interested in for this, and they're located, as you can see on the map. Then there's going to be 2 sort of what I'm calling sub-arrays in the nearshore environment. The first is up here in the north, and it's I've chosen this location because it's the nearest sort of stretch of nearshore reef to the pinnacles themselves. So if any predator is going to make a jump from the pinnacle to a nearshore area of habitat, there's a fair chance that it's going to be right here. And then I've got a slightly larger array that I'm locating further south near the research centre, which is 7 overlapping sites, which will be able to get some detail about home ranges of particularly the bluefin trevally. Sample sizes, the 2 species, because the Melanopigus are found on the bluefin trevally are found on the nearshore and the pinnacles. I'm doing a sample cohort both on the pinnacles and the coastal reefs, so 15 of each. And then for the Spirina caney, the chevron barracuda, because we only really find them on the pinnacle reefs, and they are, as I say, some pinnacle specialists, we're sampling 10 on each reef, on each pinnacle reef. And that's mostly from a resource perspective. That's as many tags as we've got our hands on. Okay, in terms of designing the array of receivers, there's a few steps to go through. And the first is to work out what the range of the transmitters that you're going to use as the tags on the fish, what their range is. And we did this in 2019. We picked a particularly inclement weather day so that we've got harsh conditions. And we basically go out and we put a range testing tag in the water and just move away from it and work out how far away we can detect it. And for our tags in poor conditions, so it gives us a conservative estimate, that is, what we found was it was about 400 meters. So that gives us a conservative estimate of range for these things. There's another step that we've got to do, which is working out the efficiency of detection with the receivers, and that's going to be conducted hopefully later this year. Okay, so this is a zoom in on the subarray that's located to the south, a slightly more detailed one with 7 receivers on it. And what you're looking at here is, north is off to the left now, so everything's spun around 90 degrees. But each one of these circles represents the range of a receiver, so 400 meter radius, around an individual receiver that's already been deployed on this network of coastal reef in Kimby. And so we've tried to design it so that there's some overlap at each stage, so that any fish that we tag in this array, as it moves up and down this patch reef, habitat, we can hopefully detect it moving from one receiver to another and get an idea of their ranges. Okay, putting the receivers out themselves. So these things got to go out vertically in the water column. So we deploy them with an anchor system to the benthos and then a float to keep them upright. Each receiver's got a 69 kilohertz hydrophone on the top, which detects unique pings from the coded transmitters. So these are the things that we actually put in the fishes themselves. So here's an example of the ones that we're using, a V13, 13 millimeters in diameter. And each transmitter broadcasts a unique code, and each code is codes for unique fish. So we can tell each fish as it moves around the array. Okay, in terms of catching the fish themselves and getting the data, we basically catch these guys on rod and line from boats. We bring them into a boat, and if they're a suitable size candidate for the study, then we anesthetize them using an MS222 solution. And then we conduct an onboard surgery where we put the transmitter into the intracoelomic cavity, so into the abdominal space of the organism. While we're doing it, we take measurements of as many measurements of the fish as we can, and we get some fin clips just so we can do DNA work on these guys down the line. And then once we're done with the surgery, we can move into the recovery phase. So we put the individual into this release pen that we have on the side of the boat, and we give it about 40 minutes to an hour just to sort of recover from the anaesthetic, and we just make sure that it's in a fit state to release to the back into the environment. So once we're happy that it can be released from the environment, then we let it out of the release pen. And this is just a short video that we took last year doing this on a trevally species in one of the nearshore arrays. So this guy recovered fine from the surgery, and as you can see here, he's got a dart tag in the dorsal musculature, so that if we catch him in the future, we can recognize the fish. And then once they're back out in the environment, we just have to wait and wait for them to move around the environment and broadcast to these receivers, and then we can collect the receivers in. So periodically, I'm not sure when I'm next going to be able to retrieve these guys in. We can go out and grab them back in out of where they're located. We bring them back to land, and then we can download any detections that they've made into this software that we get from the manufacturer, which comes down like this picture on the left. That then gets converted into a CSV file, and then we can export that into R and use packages like VTrack or just other code that we can write to analyze movements and the raw data. The kinds of results that we're after really are looking for things like site fidelity and residency, which will help me answer the questions about whether these organisms are tightly connected to the pinnacles or whether they roam off elsewhere. And we can get information about periodic movements, such as DL movements, and this figure on the right here is showing individual fishes moving in a day and night pattern. And then hopefully things like home range sizes, and really is sort of hoping that maybe we'll get some ideas if numbers of individuals are converging in one place at one time, we might get some idea about aggregation dynamics. Okay, so that's chapter 3. Moving on to chapter 4 then, we're going to be dealing with some of the ontogenetic and life history connectivity and some food web analysis stuff. So, and again, a number of research questions. First is whether the diet of pinnacle specialists reflects their tight connections to the pinnacle. So if you remember the idea about these, maybe these seemingly specialist barracuda that stay on the pinnacles, maybe they move off at night and feed elsewhere and bring energy subsidies back to the reef. In contrast, is the diet of wide roaming habitat generalists, is that reflected in the wide range of feeding? And then the third, more complex question is whether the historical diets of some of these individuals reflect ontological migration, so whether they're feeding as juveniles. Can we pick up any of that information? And so the approach is basically to use stable isotope analysis to understand carbon-13 signatures, the ratios of carbon-13 isotopes in these organisms. And first we identify some potential primary producers, which in this context we call source end members, because not all of these things are actually primary producers. We have things like detritus right here, which detritus can form the basis of a food web. So in this context we call them source end members. And we're going to conduct this again on the same habitat types, pinnacles, near shore and offshore. We also take a bunch of predator species and take some muscle tissue from which we can infer adult diet. And then we're going to look at some of the otolith cores, which are formed while they're juveniles and while they're feeding as juveniles, so we can infer juvenile diet. And then we use a compound-specific stable isotope approach to dig into some of the more detailed components of that. And that's basically just looking at the differences in essential amino acids and their signatures. All right, so in terms of study design and layout, once again, we've got our 4 pinnacle reefs. We're also pairing these up with 4 nearshore reefs that we already work on and 4 offshore emergent reefs. So these are also offshore like the pinnacles, but these are the guys that come up to within a few meters of the surface. And across all these, we've got 9 different source end member types, 6 of which are specifically going to be marine and 3 terrestrial. And then the predator fish species, of which we've got 3 species, 2 of which you'll be familiar with. So the primary producers, or source end members, as we're calling them. So we've got things like plankton and macroalgae, epiphytes that occupy macroalgae as well are getting included, detritus that we can get at using some of the detritivores, zooactinian coral and seagrasses. So these are our marine candidates. And I'm also really interested in these 3 potential terrestrial source end members. And that's because I'm hoping to be able to investigate some of this cross-habitat connectivity that I mentioned before, and whether any of these guys are supplying energy into the marine environment from the terrestrial. And so that includes things like mangroves, oil palm, which is incredibly abundant in Kimby. There's a lot of oil palm plantations there, and river water runoff. So far, I've already conducted a bit of a pilot study on 2 source end members from Kimby Bay, from the nearshore, offshore and pinnacle and that's both macroalgae and plankton. And we're seeing some broad scale differences between these M member types in terms of their carbon-13 signature, as you can see here on this diagram. It is unfortunately difficult to detect differences between the sites. So for plankton here, whether we can tell any difference between nearshore, offshore, and pinnacle, which is potentially problematic, but that's kind of where the compound-specific approach may come in useful, although it's an expensive approach and or time-consuming. But we also have a number of mixing model approaches that we can use to also try and tease out some of this stuff. So data collection for the predators. And again, it's these two mobile predators, the bluefin trevally and chevron barracuda. But now I'm also including the blue-spotted hind, or Cephalophilus cyanostigma, mostly because I want to be able to compare this guy, because he's more of a resident-type individual that tends to hang out in the same spot, and use that residency as a contrast to the mobility of the other two. Okay, how it's all going to work in the field. I'm going to extract muscle tissue samples either by spear probe or from lethal sampling once we've captured them on rod and line. In that instance, we'll also take the otoliths. Tissue samples are dehydrated in the lab in Kimby and the otoliths extracted and cleaned. And then it's all returned to JCU for processing. Once we get it all back to JCU, the muscle tissue samples get homogenized in the molecular lab, and the otoliths are probably going to get pooled, because at this stage I'm not quite sure how much mass I'm going to get out of one core of an otolith. So once I can get an idea of that, I'll work out how many otoliths I need to pool together, and then they'll be homogenized and sampled together. Then all of these samples will get subsampled and sent off to the Advanced Analytical Center in Cairns for mass spectrometry, excuse me, and we'll retain subsamples in case you want to do compound-specific analysis on the amino acids later. Okay, the sort of results I'm after in terms of data analysis. So once we get the raw data back from Cairns, from the mass spectrometer, we can then use some mixed modeling approaches using SciR, which is a package that we can use in R. And that allows us to basically identify differences in end member signatures across the board. So whether these source end members are actually differentiating out in isotopic space. And then once we can understand that, we can relate those patterns to the same patterns in different predators across the different habitat types. This is an example from a paper 4 years ago using this exact same approach, using principal component analysis. And then from that, we can infer some information about ontogenetic and trophic connectivity metrics. All right, that's the end of my chapters. Just a bit about progress and planning. So far, there's a bit of progress we made in 2019. That's my first 10 months of 9 to 10 months of my project. So we've got animal ethics at the beginning of the year. Managed to get all the equipment pulled together in JCU and then shipped to Kimby. We got the array deployed between April and June last year. And then we started tagging the first cohort of the bluefin trevally. We also managed to get the first batch of predator stable isotope samples back here, homogenized, prepared, and sent to Cairns for analysis. And I obviously got all that, the source end member data back, and I've been running some preliminary analysis on that. And I've also managed to complete my HDR induction. Publication plan is is essentially just putting each chapter to a publication, and I do want to try and convert the general conclusions into some kind of recommendations for management as well. So that's the publication plan as it stands at the moment. Funding and budget. Been pretty lucky to get a few little grants to help make this all happen. So we got some money from the SeaWorld Rescue and Research Foundation, and also from CSIRO. Most of that is able to cover the final budget bill that you can see down at the bottom, the funding bill. And yeah, so on to time management. So this is all clearly now, given the current scenario, needs to be taken with a slight pinch of salt. Here we find ourselves in April 2020, and I had 2 fieldwork seasons built in for this year. with more happening down the line. And probably these are all going to have to get bounced back. But I do have a sort of I've worked out how this all has to sort of interact with each other and move from one to the other. Which brings me on to risk management and the dreaded coronavirus that's going on at the moment, which is basically my number one risk for this project. Clearly, it's impacting all of our work at the moment, and for me, it looks like both of these 2020 trips are going to be at least postponed. Knock-on effects of that are battery drawdown on all the telemetry gear out there, and other knock-on effects on my timelines. I've got a few things I can work on in the meantime, so I do have some existing data, particularly on some distribution and abundance of predators, which I can sort of incorporate and work on as a sort of separate standalone piece of work that might feed into this stuff. I've got those stabilized tape results that should come back from the pilot study, and I'm kind of eyeing up the idea of maybe getting the Perduraean samples from the GBR, so I can start working on the microsat development. But that's just an idea at the moment, and then hopefully seeking some kind of extension to get all this work done. But I am open to ideas. Okay, that's it from me. I just have to make some thank yous to my advisor panel: Jeff, Mark, Hugo, and Brett, and a whole heap of organisational support from JCU and the Centre of Excellence, the Australian Research Council and Discovery Programme for funding my scholarship in the first place. Obviously, I'd like to thank SeaWorld and CSIRO for the funding from there. Cooperation and partnership from Mahoney Nadari and Walindi and PNG, and obviously, of course, the Jones Lab and the rest of my centre chums. And then, obviously, last but not least, my lovely wife, Gemma Galbraith, who is always there for me. That's it.